Good afternoon. So back to basic science. So biomineralization, the biogenic formation of mineral deposition, is one of the most widespread process in nature. The key feature of, if you look around, the key feature during the development of nacre, the shell, or teeth and bone, is that the crystals are all uniformly sized and they have a crystallographic orientation. So my lab is interested in looking at how bone and teeth calcify. So if you look at the pie chart, you can see that the composition of bone and dentin, most of it is the mineral, which is calcium phosphate. Then you have water, which is necessary for directing the crystal orientation. And among the protein matrix, collagen forms the predominant protein. And there's a small part in the pie chart called the NCPs. These are the non-collagenous proteins. And it is these proteins that we are interested in because they are the ones that are responsible for hydroxyapatite nanocrystals, which is a calcium phosphate. So bone and dentin is a matrix-mediated mineralization process. And what the cells do is they secrete collagen. And this collagen, type 1 collagen, self-assembles to form a template. And they do so by staggering with one molecule against each other. And they create this hole and the overlap zone. It is within this whole region that the NCPs bind, and they specify the site for mineral nucleation. So it is this unusual combination of hard inorganic material and the elastic collagen matrix that gives bone and dentin its superior mechanical properties. Disruption or mutations in either collagen assembly or the non-collagenous proteins leads to several defects like osteoporosis and dentinogenesis imperfecta. In the search for NCP that was responsible for calcified tissue formation, we identified a protein from the dentin matrix, and we named it as dentin matrix protein 1. Looking at the sequence, it has the acidic amino acid residues for calcium binding, and we identified apatite initiation domains as well as collagen binding domains. You knock out this gene, you have dwarfism and chondrodysplasia, and you overexpress this gene. The transgenic TG stands for the transgenic mouse, which is overexpressed. You have ossification of the cranial suture and ossification of the carpal bones. So it's an accelerated when you overexpress DMP1. And in order to determine the structure of the mineral, we used DMP1 and did calcium phosphate nucleation studies, and you can see oriented crystals being formed. In the presence of collagen, they do have needle-shaped crystals. We also use this protein to regenerate dentin, and we can see that the new dentin formed integrates with the old dentin. And recently, we have used the pulp stem cells along with DMP1 and the collagen scaffold. And not only do we regenerate the mineralized matrix, but we can also form vascularized tissue. And this is something that we are currently exploring. Thank you.